And there's a number of reasons why you use genome sequencing. You're getting more information because you're looking at a whole genome rather than a few genes. And as I was saying before, you get things like horizontal gene transfer, which this can take into account more than if you're just looking at multi-locus sequence typing genes. And well, I'm going to discuss in a second, but people have tried to design control methods based on genome sequencing. And so the premise is that if you have an outbreak in a hospital or something like that, you can do whole genome sequencing of the different sequences or the different strains from that outbreak and try and figure out what the source is. So recently there was a case in Birmingham where there were burn wound victims, and so because they have wounds all over their body, they're really prone to getting infections. And these people kept getting the same infection. They kept getting infected by something called Pseudomonas. And they were doing these deep cleans of the wards, and they couldn't figure out like where this was coming from because people kept getting it and they were thinking, maybe it's the staff and all this kind of thing. And so a group of people went in and they swabbed the burns victims, the people that actually had the infection. They took swabs from the staff and also just from around the ward itself. And they actually found that there was a valve on one of the taps that has to maintain a 36 degrees all the time, the water there. And they found that there was a biofilm on this tap that actually had this particular strain. And so by actually making this into a tree, they could see the source of this outbreak, these repeated outbreaks from doing this. So that's the power of whole genome sequencing, is that you can actually trace this back to it. So it's pretty cool when you can do stuff like that. But there's an example here where there was an outbreak in a hospital. So it's in a neonatal hospital. And you can see here, so these are babies. And this is time. And you have babies carrying MRSA in this case. And the grey areas here are times when there was no MRSA found in the ward. So they're constantly like swabbing the babies, because you can imagine in like an intensive care unit in a neonatal ward, you don't want babies getting anything like MRSA. It would be really, really risky. So they're constantly swabbing them and checking them, and then they found these MRSAs from screening in these babies, and this is how long they had it, and these are the number of days when that particular ward didn't have any MRSA. And so from these swabs, when they found it, they actually sequenced it. And so you can see here, this is another kind of tree. And these are your different patients down the side, and this is how related they are. So in this is the number of SNPs, so number of single mutations difference. So you can see a lot of them only have one mutation difference from this P5 individual, which is this one here. And then some of the other ones have, what's that, like maybe 5 or 6 SNPs. There's some kind of gradient here. And so they took the test a bit further, and they actually looked at people that were in the maternity ward. So this could be mothers, partners of the mother as well, and people that were screening as negative. So these are babies in the ward that were being screened as negative for MRSA. And they showed through whole genome sequencing that some of the babies were transmitting it to their mothers, or in this case, their mother's partners or their fathers, presumably. So you can actually trace these transmission events. So in terms of the number of SNPs. So if you look here, you've got P8, which is this purpley coloured baby here, and then that's the baby's mother, and then that's the partner of the mother. So you can see this transmission, and if you look at it on a phylogenetic tree, you can see that they're quite clearly connected. And it's the same, you can see these transmission events by the colour, and they correspond to this. But they were quite interested into why MRSA was coming back into the hospital, so they were thinking, maybe the transmission is going the other way as well. And whilst they were doing this, they were also taking samples from hospital staff, which is this big H here. And they showed that, so all of this is from one individual. So one person, one member of staff, and you can see all of her strains are really closely related. And right in the middle of there's one of the baby strains. And so they actually showed through whole genome sequencing that this member of staff, even though the ward, as in none of the patients had MRSA during that time, this member of staff brought MRSA back into the ward and actually infected this child. And they think it probably infected this child because the mother was found to be carrying MRSA, even though the baby didn't. So the baby probably transmitted it, even though the baby was clear of MRSA. So I think you can see that whole genome sequencing can be quite powerful. And actually, as a result of this study, it changed the way that they handle MRSA in the ward and how they try and control for it, because it's clear that, you know, it's actually on members of staff as well. So you're going to do something similar, but on a slightly different data set. So if you go into practical 3, we're going to look at a neonatal intensive care unit and look at the Staph aureus outbreak in that.